The light reactions of photosynthesis will be taking place in the thylakoids specifically. These light reactions will be collecting the light energy and packing it away into a usable form that the Calvin cycle can then reduce the carbon dioxide. The actual places where the light reactions will occur are what are called photosystems. A photosystem is simply composed of a whole bunch of pigment molecules assembled together around this central pair of chlorophyll A molecules. Now, these molecules or these pigments that are in the middle will always be chlorophyll A's. Chlorophyll is going to play a special role in photosynthesis. But the pigments that will be surrounding that chlorophyll A pair will be a whole assortment of pigments. Some of them may also be chlorophyll A's, some of them may be chlorophyll B's, and some of them may be the carotenoids that we talked about. So we've got a whole assortment of pigments that will be scattered around, and the whole job of those scattered pigments is to harvest light energy. A lot of times those pigments are called antenna pigments, and they're called antenna pigments because they are acting like a little antenna. We've got them all spread out, and this will hopefully maximize light interception so that when light comes in, that photon is going to hit one of them, and hopefully that pigment will be able to absorb that light energy. Once that energy is absorbed, the pigment's job is to pass that energy around. That's being shown in this picture as the black arrows. Here we are simply passing energy, not electrons, but energy, to ultimately this chlorophyll A pair. So the job of the antenna pigments is to collect the light energy, transfer it to the chlorophyll A pair. And they will all continuously work at that job, funneling all of that energy that they've collected to the chlorophyll A pair. So we've got lots of energy coming in right here. When the energy comes in, this is when we will have an electron that absorbs that energy. It gets boosted up to a higher energy level, and that would be when it's at that very unstable state. Once it gets to that very unstable state, we will have a molecule whose whole job, and that's this one right here, is to collect that electron in the excited state. So the primary electron acceptor is going to grab that electron as soon as it is boosted up to that higher energy level. We have these photosystems scattered all throughout the chloroplast, all throughout the thylakoid of the chloroplast, and we do have them functioning in slightly different ways. We have two different photosystems. These photosystems are named in the order that they were discovered. So Photosystem II actually does function first, and Photosystem I is going to function second in the process of photosynthesis. These photosystems, they have slightly different roles. They do also have slightly different pigment collections, and this is going to change their ideal or best wavelengths slightly. So sometimes you do see Photosystem II referred to as P680, and Photosystem I is sometimes called P700. Now both of those photosystems do have that overall structure that we just looked at, where you have a chlorophyll A pair of molecules surrounded by a lot of antenna pigments that will be collecting the energy from the light. Additionally, they're all going to have that primary electron acceptor that will harvest or grab that electron as soon as it is boosted up to a higher energy level. Now what happens to that electron that is grabbed or boosted up to the higher energy level is going to vary depending on whether we're looking at Photosystem I or whether we're looking at Photosystem II, or what type of electron flow we're looking at. We do have two different types of electron flow inside the chloroplast, and this is non-cyclic or linear electron flow, and then we also have the cyclic electron flow. The primary pathway that the electrons are going to take is going to be the non-cyclic electron flow. So we'll describe and discuss this first one, non-cyclic, initially, and then we will cover the cyclic electron flow. Now, the non-cyclic electron flow is taking place in the thylakoids. So we are going to have the thylakoids here. We will have our photosystems embedded in those thylakoids. And with non-cyclic electron flow, it does begin with Photosystem II. In Photosystem II, again, same arrangement as what we had before with our photosystems. We have lots of pigment molecules scattered out so that when the light comes in, hopefully it will strike one of these pigments. So when it comes in and it strikes a pigment, that energy is absorbed, transferred around until it gets to chlorophyll A. Chlorophyll A is going to boost an electron up to an excited state, and that electron will then be collected by a primary electron acceptor. We would like to have more energy come in and get transferred around and go to the chlorophyll A pair, and then even more energy and transfer it to the chlorophyll A pair. In this case, we're hoping to continually boost electrons up and allow that primary electron acceptor to grab them. Well, since we intend to continually pass electrons to the primary acceptor, we must have a way to replace these electrons that keep leaving. This is where the water comes into play. So right here at the very beginning of photosynthesis, the water molecule will get oxidized. So there will be an enzyme right here associated with Photosystem II that is able to oxidize the water molecule, actually split it open, collect the electrons from it, and give those electrons to chlorophyll A. As a result of that process, we will get oxygen gas released. So our oxygen gas is a waste product, and it just comes about because we are getting electrons from the water. Now if we go back to our electron, which is now at a very high energy state, that electron is going to be passed down an electron transport chain. That electron transport chain works essentially the same way the electron transport chain worked in respiration, meaning we're going to take the electron and pass it to another molecule, which will also be in the thylakoid membrane. And when that electron is passed to the next molecule, it will drop in energy. So energy will be released. Then we will pass it to the next one, and another small amount of energy will be released. This electron transport chain, not only does it pass the electrons in a similar fashion to what we had in the mitochondria, but it is also going to do the same thing with that energy that is being released. So the electron transport chain will be moving hydrogen ions, across the thylakoid membrane. It's going to move them across the membrane to basically produce a hydrogen ion gradient. That gradient will be used for the same thing that it was used for in Chapter 9, which means that it's going to be used to power some ATP synthesis, and this will be ATP synthesis by chemiosmosis. This means that there will be an ATP synthase enzyme, similar in structure to what we had in the inner membrane of the mitochondria, and operating in the same fashion. So this ATP synthase enzyme is going to allow the hydrogens to flow back across the membrane by facilitated diffusion, and when they flow back across the membrane, it will produce ATP as a result. Now we'll come back and talk about that process of chemiosmosis and the electron transport chain a little bit more in a moment. But when these electrons get to the end of this electron transport chain, they will have been drained of any energy that they had accepted from the light. This brings us to the second photosystem, which is going to be Photosystem I. Photosystem I is also going to absorb some light energy, and when that light energy is absorbed, it will be transferred to the center of the photosystem, to the chlorophyll A pair. The electron will be boosted up, and that electron will get accepted. This time when the electron is accepted, we replace the deficit in electrons using this one that just came out of the electron transport chain. So we do not have a need to split or oxidize another water molecule here. The electron that is boosted up is going to get passed down a very short electron transport chain, And at the end of this electron transport chain, it goes to NADP plus to make NADPH. So if you look at what we've done here, we've essentially taken an electron, and this is an image from your book. We've taken an electron that was a low energy electron in the form of water. We have some light energy come in, and this electron is boosted up in energy. The energy is then drained out of that electron and used to make some ATP, at which point the electron is going to be boosted back up in energy and then ultimately pass to NADPH.